Good day, everyone, and welcome. I'm Jack Van Horn from the University of Virginia. Please allow me to welcome you to this edition of our 2020-2021 Foundations of Biomedical Data Science Seminar Series. This lecture is supported by the UVA Integrated Translational Health Institute of Virginia, the UVA Brain Institute, the College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. It is my privilege and my honor today to welcome our speaker, Dr. Giselle Petzinger, Associate Professor of Neurology from the Keck School of Medicine at the University of Southern California. Dr. Petzinger is a physician and board-certified neurologist specializing in the care of patients suffering from Parkinson's disease. She divides her time between clinical care for patients and active laboratory research. The primary focus of Dr. Petzinger's laboratory is to better understand how to promote brain repair resulting from neurological syndromes or following injury, and to translate these into new therapeutic treatments. Dr. Petzinger has been involved with numerous federally funded research projects from the NIH, the Department of Defense, and notably she serves as the principal investigator on the U.S. Army research grant entitled Behavioral and Pharmacological Enhancement of Neuroplasticity in Basal Ganglia of the MPTP lesioned mouse and in non-human primates. Dr. Petzinger's lecture today is entitled Investigating Mechanisms of Exercise-Induced Neuroplasticity in Cognitive Motor Systems in Parkinson's Disease, Translational Studies in Animal Models of PD to Clinical Research. As always, we are streaming this lecture live and for recording via YouTube. If you're watching via YouTube, thank you so much for joining us today. Also, our specially selected 2020-2021 Biomedical Data Science Innovation Lab participants are encouraged to submit any questions for Dr. Petzinger via the chat feature in their Zoom sessions. I'll synthesize these questions and I'll ask them on your behalf in the last 10 minutes or so of the lecture. And with that, welcome, Giselle. We are so excited to hear your lecture.
Excellent. Well, thank you so much. I'm thrilled to be here. And thank you, Dr. Van Horn, for the invitation to participate in this seminar series. So I get to talk about something that's obviously near and dear to my heart, which is Parkinson's disease. I'm going to just go through really quickly what we're going to try to get through in this hour, leaving 10 minutes for some questions at the end. I'm going to introduce Parkinson's disease and then talk about the field of neurorehabilitation and how that was brought to the field of Parkinson's. We'll then talk about neuroplasticity in the context of exercise and use our animal models as an example of understanding these mechanisms. We'll then dive into a very particular problem in Parkinson's, and that is cognitive problems and how exercise may be helping that. And then we'll get into some mechanisms at the end that we're basically continuing to investigate and are part of our current study. So Parkinson's disease is considered a neurodegenerative disorder. And what that means is that there is an area of cell loss. What I'm showing here is a normal brain and a Parkinson's brain. And what we're pointing here is the substantia nigra pars compacta, which are contain melanin. So you can see them as a dark matter in the midbrain. And you can see these tracts that go up and reach into what's called the basal ganglia, the caudate and putamen. And in Parkinson's disease, we know that we lose about 60% of cells, 80% of dopamine, which starts leading to the problems that we see clinically. Parkinson's disease is characteristically thought of as a motor problem. And what I mean by motor, I mean things like gait and balance dysfunction, slowness. Tremor is not necessary, although people often think about Parkinson's as tremor, but certainly not necessary. Probably the most important thing, though, is gait, balance, and slowness. But we also touch upon some of the non-motor features as it's relevant to some of the studies that we're doing. And the big one that I'm going to spend time talking about is something called cognitive impairment. I'm just going to point out that cognitive impairment in terms of dementia, Alzheimer's is the most common form of dementia. The second probably is vascular. The third is Parkinson's disease. Cognitive impairment is actually very common in Parkinson's disease and can be seen in about 30 to 40% of people within the first few years of what we call mild cognitive impairment. Common question people ask, how common is Parkinson's? Well, it's 1 in 100 individuals over the age of 50. Prevalence is about 1 million in North America. So moving on, let's talk about another aspect of Parkinson's disease that is not as well recognized, or at least not thought about, but it's going to be very relevant for the talk that I'm about to give. Because one of the challenges that we had, and I'm going to kind of walk you through what we went through initially when we were thinking about what we say exercise-induced neuroplasticity. The idea was that we were very interested in thinking about it as a circuit change, right? So obviously people get interested in knowing what it does for cells, for sure. But the idea here was that Parkinson's disease is actually more than a cell problem. And that's the other thing to really emphasize as we move forward. And I'm just showing you as an example, two particular circuits. These particular circuits called the motor somatosensory circuit and the prefrontal striatal circuit are both circuits that are involved, for example, in motor movement. And the reason I'm pointing them out is what people don't understand or realize is that this particular circuit, the middle circuit in yellow, is another term we use for this circuit is called the automatic circuit. This is a circuit that's practiced, that over time becomes obviously better, stronger connections that actually allows us to move through space without having to think about it. But we also have this other circuit that is involved in processing movement through space. It's also used for dual tasking. This is a circuit coming from the prefrontal cortex of the basal ganglia. And this allows me to update information about movement through space. I'm highlighting this just to keep in mind as we move forward and think about what we're doing with exercise. The idea is that in Parkinson's disease, people actually have deficits. The bigger deficits is in this automatic circuit. So people start requiring more demand on this prefrontal cognitive. So a lot of what Parkinson's patients undergo when they're suffering from this slowness is really a problem in automaticity, meaning they lose automatic movement. So they look and appear slow, and they have a higher demand on this cognitive process. Now, this is going to come up, as I said, again in our discussion, and the reason this is so important is that this is one of the reasons why people think there could be some early cognitive changes. Obviously, dopamine depletion could be contributing to this, but there is this other idea that maybe with this higher dependency on this prefrontal cognitive circuitry, maybe there's some sort of saturation that is making these patients more vulnerable. Okay, so the take-home point here is, obviously we know Parkinson's disease is fundamentally involved in as a neurodegenerative disease with dopamine, cell loss, etc. What I want to, again, take home point, it's also a circuit problem. And so this is going to be important as we move forward. Now, the next slide I'm showing you is kind of the idea of something that we're all appreciative of, which is called neuroplasticity. And this occurred in the decade of the brain, where people were recognizing the importance of neurogenesis that was ongoing as even in aging. And also basically the idea that experiences that we have in our day-to-day life impact how our circuits are formed. And we're going to highlight some specific ideas of that term again. And we're going to come back to these terms, but I'm going to define them a couple ways, because these are the sorts of ways that we think about neuroplasticity in the brain. I think the first point to make is very important, and that is fundamentally, it is how connections are formed. Brain reorganizes itself, forming new neural connections that are happening through life. That itself is a natural aspect of what we call neuroplasticity, circuit formation. The other thing to keep in mind is fundamentally this is happening at the level of a synapse. So we're thinking about synaptic structure and function, because both of them are relevant when we think about plasticity. The other thing that's also important to keep in mind, and this comes up when we think about things about how things are changing in relationship to other parts of the brain, which is something called homeostasis, which is that neuroplasticity also probably underlies aspects of the brain trying to reach a new normal. So if there's one area of the brain that's injured, there's another brain that's part of the brain that may be taking up the slack. I would argue that what I just described with Parkinson's disease is an example of that. So they lose automaticity, they have to think more about movement in space, and that's a form of homeostasis. Okay. The last 2 points are going to be particularly relevant when we talk about exercise, because again, very fundamentally, neuroplasticity is the way that we encode experiences. It's the way we encode new behaviors. And in essence, it's a way that the damaged brain, we can basically have the areas of damage recover in response to rehabilitation. And the field that really took that notion and really began to use it practically in humans really comes from the spinal cord injury field and actually the stroke field. So Parkinson's disease came much later to this idea. And by later, I mean in the last 15 years. So this is another slide. I think it's, just a good slide to remind ourselves that obviously we recognize how important experience-dependent neuroplasticity is in children. We see these in athletes, so they, at the top form of fluid movement through space. But really, where we're at now in the field, very much so, is the idea that we can harness these experiences and provide them in a very deliberate way to try to kind of direct plasticity. So we're trying to see areas of the brain that we think are compromised. How can we use exercise as a repair system, and importantly, understand those mechanisms that underlie that repair so we can tap into that as well. So when I started getting into the field, so again, this would be the early 2000s, that long ago. It doesn't seem that long ago, but it's been that long. Basically, one of the things that we started seeing in Parkinson's disease was this idea that we could do more tapping into gait and balance problems. And so most of the original work from Parkinson's disease really came at trying to recover gait and balance. And the reason I'm saying that is because I wanted to, again, the idea of experience, that these physical therapists and these particular exercises that were being delivered were really trying to tap into some of the more fundamental points of Parkinson's disease. And this approach to the field that we had was very different. I'm going to juxtapose it to the aging field. Aging field, which we'll talk about in just a second, was more interested in aerobic exercise. But because of Parkinson's disease, the fundamental problem being with gait and balance, if we're thinking about neuroplasticity, then you go after recovery of that through these different types of exercises. And one of the things that comes up often about thinking about all these different exercises, again, what do they have in common? Why do many of these different types of exercise show the same benefits with gait and balance, which is what they've done? And one of the ideas that started coming out of this discussion about all these different types of practices that could lead to improvement was that they had basically a practice learning skill modality. Fundamentally, patients are getting repetition, they're getting feedback to get better at gait and balance. And so that what's coming out of the field then, and you're kind of getting an understanding of this approach, is in exercise for Parkinson's disease, we were very much skill goal oriented right out of the gate. And we saw some pretty amazing improvements. And I'm going to just show you an example. So this is a typical patient with Parkinson's disease. And the way, again, we characterize it is slowness and gait and balance. You can see he's taking a very short steps. And in just a moment here, he's going to take even tinier steps. We call this a context dependency problem, that they have the idea that once they change even the flooring, it gets very hard for them to think about how to get through that. And so the steps actually get slower. And so this is an example of an individual who went through gait and balance training. This was no change in medication at all. And so this sort of response is what basically the literature showed, that with practice, particularly focused on gait and balance, you could definitely get pretty significant gains in function. Okay, so this is, again, highlighting out of the gate what we became very interested in from the field of Parkinson's disease, and that is exercise interventions that were requiring practice, that were specific to the deficit, that were also difficult for the patient. In other words, we would progress them through harder and harder practice so that they would get better, right? And keeping it complex. So this was interesting because many of these thoughts that we were describing really were borrowing from the stroke field, the spinal cord injury field, and we were all talking the same language, which is the idea of motor learning. So we were coming at it really from kind of a motor learning point of view. And so when we were trying to conceptualize how are we going to study this in Parkinson's in animal models, right? So we're now taking what we're learning from humans and we're thinking, okay, what's the specific question we're going to ask now about why these behaviors are better? Fundamentally, we started with the idea of learning, and we talked about that already. So the idea of learning, of course, is going to happen right at a synapse. So we became very interested in really tackling this question. Do we see evidence for synaptic changes, synaptic plasticity in the basal ganglia, right? And then the second factor, not a small factor, was kind of all those other things that are beneficial coming in from the body to the brain, right? And the aerobic field was really taking off on this, where the idea is that blood flow, just, get your heart rate up, get your body in shape, and all these factors would somehow make it to the brain, and then your brain would be better. So we recognized that there were sort of these juxtaposed ideas that were coming in the field of exercise, and we were trying to reconcile that in our brain about what does this really mean. And so the idea we had come up with was that the idea is that something is happening very locally within the brain in those areas that are having problems, deficits. Basal ganglia is an example of that, right? I pointed out about the problems in the automatic circuit as an example, prefrontal circuit as an example. But at the same time, these factors that may be coming in from the periphery may be providing the environment, right? So that if this repair is going to happen synaptically, what may be coming in from the periphery is support, so a type of a fertilizer, if you will, right, that may be supporting these types of synaptic changes. Okay, so this is sort of another way to think about it. So we were sort of here on the left with Parkinson's, much more skill, and here's the heart field, right? The idea of the aerobic exercise field, the aging field that was really tapping into the idea of blood flow. All right, so we'll get back to that in just a minute. I'm just going to go on, and we're going to talk about animal models, our little joke here on translational, and HIPAA protection for our mouse and our people. So here we have an example of some work that we have done. Again, this is in 2007. Basically, we started off with an MPTP rodent model. Now, there's a lot of confusion in the field about how you design some studies with MPTP models. One of the things I wanted to point out here, because this is going to be important, we know after we give MPTP, so if we lesion an animal, we know when all that cell death is going to be done. We actually know that by 5 days, cell death is gone. So if I'm using an intervention out here, which is what we did, we're not looking at neuroprotection. This is not a neuroprotection study at all. This at this point is looking at a model in which there's already cell death and there's dopamine depletion. So think about it this way. I'm looking at a Parkinson's patient, okay? So I'm not trying to prevent cell death. I'm trying to see in this animal model that has already have deficits, can I do anything to that brain now? So this would be a neurorestorative model, okay? So with that in mind, we have these two groups of animals. These are a Parkinsonian MPTP animal that I said have completed its cell death. So we know already they're Parkinsonian, and here's our healthy control. And we basically have them in exercise and no exercise groups on both sides. Okay, and so this is basically some of the original, some of the original treadmill work that we did. And you can see that these animals actually get up to pretty high speeds. Now, you can argue that most of this is aerobic, which is true. But if you don't know a mouse, obviously most mice are not running in a direction like this and certainly not heading towards a wall. So we did feel that this animal obviously had to have some prefrontal circuits activated here to be able to attend to the task and concentrate on what it needed to do to make sure it didn't hit the back end and get zapped. So there is aspects of cognitive, certainly attentional control. What I want to show you here is basically the original paper that was really a paper focused on neurorestoration. There had been already one other paper out there using a similar model to us who had looked at neuroprotection. So again, just to make these juxtapose these two different ideas. In this case, they're Parkinsonian. So what I'm looking for is improvement in behavior. And what we see here is after we have had the animals run for about 4 weeks, weekends off, we see that these animals get better. So here in the dark black is sailing, here's the MPTP animal. And so here we see in the exercise group that Parkinsonian animals, in fact, get better. And it's kind of like no surprise now. If I practice skill, I'm going to get better at skill. And we're certainly able to demonstrate that in a mouse. But what was surprising to us were a couple other things that came out of that. And one of the things that came out that we thought was so important was when we looked at total dopamine levels. So we talked about dopamine and how dopamine is depleted, right? So we can see that in this MPTP animal. Here's the black bar. Here's your dopamine depletion, our Parkinsonian animal and our control. You can see it's quite depleted in the mouse. What's surprising to us, though, was when we saw exercise, so this is after the animal finished exercise, we didn't see any recovery of dopamine. And that becomes very important because it kind of helped us kind of secure our thoughts, which was we were interested in circuitry. We were interested to know something downstream, what was happening downstream that may be relevant for driving these effects, because the tendency was to think if I am improving in Parkinson's, I must have some element of neurogenesis, or I must have some sort of dopamine recovery. And that is not the case, at least not in this type of experimental design. And that, again, pulled us out of the Niger and now into the circuitry to start looking more specifically at what's happening there. Now, what we were able to do, though, is we were able to look at release of dopamine. And this is basically a physiological approach called voltammetry. All I'm showing you here is a slice of a brain and all these different areas of places where we introduce the needle and we excite that area and look for dopamine release. Take-home point is way down here. What we're showing you here is what's in hatched or exercised animals in all those different positions I'm showing you in the basal ganglia up here, and in dark gray is the non-exercise. You can see anywhere that I excite, I'm getting more dopamine release. So take home, not changing the amount of dopamine, but absolutely changing the properties of the dopamine, meaning that they're being released. The other thing that we saw, I'm just going to slide down to here, is to show you that we didn't see any changes in cell number. So no, there is no evidence of neurogenesis with exercise. And we've never seen that in our model. Basically so what I'm trying to say is whatever change that I've shown you so far, it's basically in release properties. This slide up here, which is just a take-home point to say that we're also seeing in dark gray is our ability to image the transport of dopamine, which is a reuptake system. And actually the transport system goes down, meaning dopamine is not being taken back up. In fact, it's being left out in the synapse. So this is homeostasis. This is a perfect example of homeostasis, and it matches the ideas of neuroplasticity. Okay, so what else did we learn? And I'll try to move a little quickly here. We also learned that we were changing dopamine receptors in the basal ganglia. D2 is a very critical one for motor control. Here's your MPTP Parkinsonian animal. We saw it go up with exercise, but more importantly was being able to translate that into an image. And what we were able to do was use falopride, F18 falopride, to be able to see whether or not we had D2 receptor changes within the striatum, and the take-home messages we were. So this is a superimposed PET image with an MRI image of a mouse. And here we have D2 falopride in red. And here we have it in a Parkinsonian state, D2 regulation or D2 expression going down. And then with exercise, D2 is up. We were particularly interested in using falopride because raclopride, which some people may be familiar with, is actually probably a better measure for dopamine amount. And we didn't want to look at dopamine amount. We were interested specifically on the D2 receptor. F18 falopride was designed by a chemist at UC Irvine, and he was able to help us execute this study. So take home point is that we see release of dopamine and we see an increase in dopamine receptors, although we see no change in total dopamine levels or cell number. We were also able to replicate this in a much smaller study in Parkinson's patients. So that study that we talked about in the very beginning, we talked about all these different types of exercises. One of the exercises that we really got involved in, which I don't have time to show you pre-post videos of that, was a treadmill study. So we basically put patients on treadmill. These were newly diagnosed patients with Parkinson's on no medication at all, because you and I both know that medication can sometimes affect some of these tracers. So we had patients on no medication. We put them through 8 weeks of exercise. And what you're seeing here is a pre-post, actually it's a post minus pre D2 scan. Here's, again, superimposed, here's your PET scan with D2. Here's your MRI, and this is a superimposed PET on a MRI image, just to show you how sensitive that falopride is to the striatum. And here's this is actually the most important picture, which is 1D. What you're seeing in red is a subtraction of post-exercise from pre-exercise to show that what we saw in animals was also true in humans. And so that, again, showing how important exercise was, and actually just changing just the local dynamics of dopamine within that striatum. So again, we're downstream of the nodal system. We're actually within the basal ganglia. Another aspect of neurotransmission that we're interested in is actually something called glutamate. For those of you who study synaptic structure and function, you know how important glutamate is in terms of regulating the functional aspects of the synapse. So I'm just showing you a cartoon drawing of a synapse that is happening within the striatum. So these are going to be glutamatergic inputs coming in from the cortex. So here we have here, and on the postsynaptic side are what we call medium spiny neurons. And that's really what makes up the bulk of the striatum. So I'm showing you a synapse, and what I'm showing you in cartoon image here is a number of very different important facets that we saw change within the glutamatergic system with exercise. And one thing we saw is in MPTP, what's interesting and what's been reported is when dopamine depletion occurs, you actually have a buildup of this glutamatergic system. It gets very hypoexcitable. We call it increased glutamatergic drive. It's aberrant. It actually leads to aberrant behavior. So we have, in one aspect, we have loss of synapses within the striatum, but in those remaining synapses, we have this buildup of glutamate. We see a lot of glutamate presynaptically. We can use EM. This was how we looked at that. EM shows high levels of glutamate here, and postsynaptically, we could see a buildup of glutamate postsynaptically, and that's what we see here in white. And then with exercise, we saw it normalize. So that's the take-home point. Glutamate, which is typically thought of as hyperglutamatergic in the Parkinsonian state, we saw it normalize with exercise. So again, further evidence that in fact we are seeing homeostatic changes, really. So we're driving, I'm going to pull through, get through this, some pretty significant players in terms of neurotransmitters within the striatum itself. And a lot of that work we ended up taking on into the prefrontal circuit as well, prefrontal cortex in particular, and we're able to replicate a lot of what I just showed you there in the striatum. So another very important point that people don't realize is here's a medium spiny neuron right here. We've blown this up. And we're going to take a look at this medium spiny neuron because what we're particularly interested in is all these little thorny things happening off to the side. These are synapses. And remember, again, as I mentioned, if you're interested in plasticity, fundamentally, you do want to get at the level of the synapse, and that's why we do these animal studies. So what I'm showing you here down below is a blown up version of this just to show you the number of contacts these medium spiny neurons are. As I told you, that coming from the cortex, the cortical striatal input is pretty big. So is the thalamic striatal input. And basically, they provide a glutamatergic tone on that medium spiny neuron. I told you that when dopamine depletion occurs, we saw changes in glutamate neurotransmission. I said it went up, but we also see loss of contacts, and you can see that. So there's actual disintegration, if you will. We see loss of synapses within the striatum. So that's such an important point because, again, I think one of the issues that often comes up is people have this idea that everything is happening down in the midbrain. The truth is a lot of the action is happening upstream at the level of circuitry. And so this is, again, just a perfect example of why we need to be focused on circuitry in Parkinson's disease. And also, I'm going to just show you one other, this is, again, another medium spiny neuron. This is work done by Bill Toy in the lab. He basically was able to trace a 3D image looking at these medium spiny neurons. And what I want to show you to the right is just examples of animals, basically a selected a cartoon, if you will, of filled cells, just to show you how different the arborization looks of these cells. So here we have a normal animal. This is a medium spiny neuron within the stratum. Here's an MPTP animal. This is dopamine depletion, guys. Look how different that looks, right? So you can see actually lots of loss of dendritic arborization right here. And here's exercise. So yeah, that's pretty important because, again, if everything holds true in the universe, this animal model is telling us how important it is that we look at circuitry and synapses. I'm not disregarding the importance of dopamine, but what I'm telling you is that this is key because it's going to give us some new therapeutic angles to go after. Okay, so how about counting spines? So this is, again, more work from Bill Toy in the lab. And all I'm trying to show you here is how we go about doing this. This is a Golgi stain of the dorsal striatum. The dorsal striatum is within the basal ganglia. It's the most important motor aspect of the basal ganglia. Here it is filled with Golgi. And so what we can do, what Bill did really, was he basically counted. He counted all these synapses. So you can see with exercise, we actually see some increase even in normal animals, which is good to know. So you don't need Parkinson's to get benefit from exercise. But here we have with MPTP, we see some stripping, and here we have MPTP exercise, and this paper came out in 2011, basically showing evidence for synaptogenesis. Take home point then, in Parkinson's disease, we have not seen neurogenesis. And this is a model where we have created a Parkinsonian animal, right? It's not a neuroprotective model. It's already a Parkinson's animal. Now, when we apply exercise to that animal, what we do see is evidence for repair, homeostasis, neuroplasticity, and synaptogenesis. We don't talk neurogenesis, at least not within the basal ganglia and nigra, not in our PD field. Now, there are some evidence for hippocampal, and people hear about that, neurogenesis, but in areas that we're particularly interested in, for us, it really is more about synaptogenesis that we're interested in. Okay, So let's change directions a little bit, and let's talk now about the different types of exercise that we kind of talked about from the beginning, right? We said in the Parkinson's field, we're coming at it from skill-based. We want to get this gait and balance effect. But the truth is there's a lot out there about aerobic exercise. And we were interested in understanding, does it matter what I do, particularly if I'm interested in getting at some of these other circuits, like the prefrontal circuit? Can I tap into that system? Because, well, it's a problem in Parkinson's disease. And Daniel Holschneider in the lab came up with a really nice way to study this. And by the way, for those who don't know what skill-based exercise, this is things like yoga, tai chi, pretty much any type of structured exercise that you can think of. And then aerobic exercise, we think of as getting more your heart rate up. So this would be examples of running, swimming, cycling, that sort of thing. And here's just examples of that, not kind of unusual examples. I don't know, we thought that was kind of cool. Okay, so here we go. So the difference here is we have animals. Both of these animals have these running wheels, and these wheels are on motorized rods. So they are definitely getting the aerobic practice without a doubt. But the difference between these different running wheels, as you can see, is one has spokes removed and one does not. And so what we were trying to do here, what Dr. Holschneider particularly was trying to do, was try to tap into the idea of how important is it to be engaged in what you're doing? Can you just be running or getting your heart rate up, or having that extra benefit? Is that something that could be relevant? And so what he did was he put those animals through those running wheel for about 6 weeks. And he has his background is being able to do cerebral blood flow mapping. He does that by using this particular dye, C14 iodo antipyrine. And so after the animals have finished exercise, he keeps them perfused with this dye. And basically, after that perfusion is done, the brain is removed, put on film, and we can do a type of parametric mapping like we do in humans. So this is looking at those two groups of animals now. You can see what Dr. Holschneider did was put a seed in one of those areas, the prefrontal cortex, to see what else is being connected to that area once that area is seeded. And the comparison is, again, between the non-skilled and the skilled. And what was kind of cool about this was that it seemed to suggest that it did matter, that blood flow seemed to suggest that, yes, the more cognitively engaged that you are in the task, you can change at least regional cerebral blood flow. And we believe that that is a marker of neuronal activity. That's part of the ongoing studies that we're doing right now, is to begin to understand what is underlying that effect, evidence for angiogenesis, and also neuronal synaptogenesis. So back to this story then, let's talk a little bit more about, more specifically about aerobic exercise. And what I decided to do, so we had already done some work with skilled exercise, and given the fact that the aerobic exercise field, particularly cognitive aging, had shown so many of these incredible effects with cognition, particularly executive function, which is what we're interested in. Prefrontal cortex is executive function, and memory, decided to go ahead and see if we could take a look at that in patients with Parkinson's disease. And so what we decided to start off with was just a cross-sectional study. So no intervention, just patients that we knew were already active were recruited into the study. This was 48 patients, and this is sort of a breakdown of their age. So about 64 years of age, slightly higher, more men than females, which is more men than women, which is actually very common. In Parkinson's disease, there's slightly a higher incidence of Parkinson's in men than women. It's also easier to recruit men in general, most studies. And then the years of diagnosis is about 10 years of diagnosis. We looked at some aspects of fitness. Our particular interest was this one. This is a way that we can look at cardiorespiratory fitness. So people talk about VO2 max. So cardiorespiratory fitness has a lot of different kinds of ideas behind it. But one idea is that it's sort of reflective of blood flow as well. So obviously VO2 max is how well I'm extracting oxygen, but people also use it sort of as a surrogate, if you will, of how much blood I'm getting into the brain. So I'm pointing out a couple aspects of Parkinson's that I mentioned before that we're interested in. I didn't particularly mean to highlight this, but memory is definitely one of them. So it is not just unique to Alzheimer's, but we've definitely seen PD. Executive function, though, I would say is quite big. Visual spatial, so executive function is that prefrontal system. visual spatial function, which is my ability to navigate through crowds, attention, processing speed. All of these are actually pretty common deficits that we can see even early in Parkinson's disease. And so what we were able to show in this, let me go, oops, yeah. So I'm just going to highlight quickly some very points on this particular study. I'm going to show, first of all, we did take, with the MRI, we were able to segment the brain and do some free surfer analysis with our patients with Parkinson's. And this is just a cartoon to show a couple areas that we saw some hits with cardiorespiratory fitness. We saw it with thalamus and pallidum, which is a little surprising, but I'm going to show you some data that may explain that. And then we also saw it with a couple of these domains. So we saw, again, in this cross-sectional study, a very significant association between executive function, episodic memory, and visual spatial. So it is lining up with what they're reporting in aging. What's different for us is the Alzheimer's field reports in structural changes in the hippocampus. So they see a relationship between cardiorespiratory fitness and the hippocampus and the prefrontal, and we saw it in the thalamus and the pallidum. And again, I'll tell you why we think that is in just a second. The other point of this paper is hopefully coming out soon in Parkinsonism-related disorders. It's being submitted right now. But the point is, what I also want to show is we were able to look at some mediation analysis here. All this is, it's a mediation analysis where we try to see whether the effects of VO2 on things like episodic memory or VO2 on executive function can be explained by volumetrics in thalamus. And basically, we were able to show that, in fact, it was explaining this. Okay, so I mentioned to you that I would say something about why thalamus. It was an unexpected finding. So what I'm going to just highlight is another paper, again, Holschneider, Dr. Holschneider and our group, who had done some studies looking at exercise and more particularly using the same blood flow technique, basically able to show that actually with aerobic exercise, we are seeing quite a bit of activation within the thalamus. And there's two areas in particular that we're seeing changes in the CM and the ventrolateral aspects. These are of particular interest because actually these areas are very much interconnected with the prefrontal cortex. Lots of data out in Parkinson's disease, in fact, about thalamic changes that occur with cognitive impairment. So stay tuned. But yes, lots now coming out more and more on how important the thalamus is and how the thalamus really works with the prefrontal system to help with executive function. And the same also is true for memory. Okay, so let me just keep going. So mechanisms that we are interested at looking at now that we've got obviously some interesting data. I've told you that there is already a lot published with balance and gait with Parkinson's disease. The cognitive field is still something up for debate. It's been mostly studied in aging, but there are a number of studies that have come out, two studies in particular with aerobic exercise, where they've done some changes with EF. Our study was different because we looked at cardiorespiratory fitness that hasn't really been looked at that way in Parkinson's disease, but we're seeing definitely some association there. So we're excited that we're seeing something similar to what's been reported in aerobic exercise as well. But the question still remains, why do we care? What is underlying these effects? How can we better understand that? And I'm just listing, a couple things that we're particularly interested in. I'm going to point out some very specific factors that we're looking at. Obviously, neurotrophic factors is way up there. But as I mentioned, angiogenesis and blood flow is also critical for us because some of the original work that was done in animal models where we saw exercise effects on the brain. This is a study that was done by Greenell and Black back in the 70s, basically showed how important angiogenesis was in animal behavior. So we're trying to look at angiogenesis as well. So in thinking about that, one of the things, so let's talk about trophic factors first. We got interested in looking at one particular cell of the brain that is probably one of the most important cells that is important for neurotrophic factor delivery, and more importantly, for support, neuronal support. And these are astrocytes. So what's kind of cool about the astrocytes is they have a way not only to tap into blood vessels, so all the small capillaries they're lying up against, but they also have very strong connections to synapses, right? And we talked about how important synapses are. So astrocytes became very important for us because we were interested to see what role they may play in providing synaptic support, number one. But number two, they're also a venue or a supplier, if you will, of neurotrophic factors. So one of our graduate students in the lab began to look at astrocytes in animals and began to look at them in these two very specific areas. We're interested in the prefrontal cortex and the striatum. And what he was able to show with exercise was this pretty impressive arborization that's happening with astrocytes just through exercise. The other thing he was able to demonstrate was, so one of the things we knew that astrocytes did was not only provide trophic support, but actually provide fuel to the synapses. And one important source of fuel, believe it or not, is actually lactate. So where does lactate come from? Well, lactate comes from the periphery. Lactate is certainly upregulated with exercise. So we wanted to find a way to see what role lactate may play in this effective exercise. So to step back for a second, if I imagine that I'm exercising, I imagine that I have some lactate buildup that's being delivered to the astrocytes. The question is, what and how is astrocytes responding to this? So here's a study that was done by Adam Lundquist in the lab, and here are some astrocytes that he put lactate on. He pulled them from the brain. And what we're showing here is just a number of neurotrophic factors that he was able to measure in these cultures. And basically what we're highlighting here are some big ones. GDNF and BDNF are very commonly described neurotrophic factors, particularly in the exercise field. As an example, we can see that actually with the administration of lactate, these astrocytes from the brain are in fact making trophic factors. So that is, again, as we try to understand how synapses are remodeling, both functionally and structurally, astrocytes may play a very important role, as we would anticipate, given its role in synaptic structure maintenance and repair. I think the other thing that was also important was that he repeated this study in a live animal. So the other study was done in culture, but he was able to show when he administered lactate to animals, he was also able to take out the animal brain. Again, he was able to replicate what he saw in cell culture, which is an increase. Here we go. These are BDNF and CNTF, GDNF, that it is absolutely, it's upregulated in the brain. And he was also able to show again in vivo study that we saw a growth in the astrocytic morphology as well. Okay, so changing tracks again. So here we are understanding what is causing repair to occur. Picking one cell as an important cell, astrocytes, one possibility is lactate coming in from the periphery, maybe helping these astrocytes not only provide fuel, so astrocytes can deliver lactate to the cell, but also play a role in trophic support. How about the immune system? We tend to think about the immune system as bad. Obviously, with COVID going on, we all think of it as bad. But it turns out, as you know, that everything has good parts and bad parts. So we were interested in looking at the good parts of the immune system. And what we decided to do was, first and foremost, was to take a look at whether or not there was any evidence for immune cells coming into the brain. I'm going to walk you through this kind of busy slide, so bear with me for just a second. First of all, we're using the mouse model of Parkinson's again. This was work done by Brett Lund in our group. And one of the questions that we were trying to ask was looking at another important cell within the brain. It's called microglia. It's equivalent in the periphery called mononuclear cells. So while the majority of, and microglia, another cell that can provide trophic support, it's also very important for repair. It is the innate immune system of the brain. What's kind of cool about this is that people for a long time felt that there was basically some immune privilege in the brain, meaning that there was no interchange with the periphery. But that idea has now gone away, and now people are accepting, yes, actually there are some peripheral mononuclear cells that can go into the brain and change phenotype to become now look like microglial cells. So the reason we did this study was we were interested in understanding, was there any contribution from the immune system, and particularly cells, coming in from the periphery to the brain in an exercise animal? Take-home point is yes. And the way this was done was basically looking at surface markers that we knew existed on peripheral mononuclear cells. And we can use what's called a FACS analysis to actually measure the presence of these cells. And so what you're seeing circled in red is our analysis looking for the presence of these cells now in the brain. The way we do that is we're going to have these 4 different groups of animals. after they complete their task, we're going to take the brain out. This is taking the whole brain out, and we're going to start, we emulsate, we separate out cells, and we start looking for these peripheral mononuclear cells, which I said we can track because we know what to look for on the cell surface. And I'm going to show you here, here's an MPTP animal that has gone through no exercise, the same model, they're going through the treadmill, just like I showed you from the very beginning. So basically 4 weeks of exercise, or pardon me, up to 6 weeks of exercise. So here's MPTP, no exercise. Here we go, 2 weeks, boom, coming in from the periphery, guys. 6 weeks, boom, even more. Isn't that cool? That's wild. Yeah, so here, and this is just quantitated. So yes, it does seem like peripheral mononuclear cells can come into the periphery. And so the whole idea that, and we toy with this not uncommonly, I mean, what does alterations in blood-brain barrier really mean? You know, again, you know, nothing is dichotomous. I mean, it's not all bad, or, you know, it's, so the idea is that there could be some important aspects of changes in the blood-brain barrier. And in this case, some aspects that are actually allowing mononuclear cells to come in. Now you would ask yourself, are you doing something bad to this brain? And the answer to that was also answered in the next slide. So what we're showing here in a little cute cartoon here is basically this is the exercise, non-exercise. And what we looked at were both pro and anti-inflammatory markers. And these are the pro-inflammatory and these are anti-inflammatory. We wanted to know whether in general, the cells that I just showed you in that brain, we can extract them and then we actually cause them, we activate them. We can use something, antigen, to basically activate these cells to start secreting what they typically make to bring it out more. And so what we were able to demonstrate in these same cells was an increase in neurotrophic factors and anti-inflammatory. So it's an anti-inflammatory markers. Here's also showing that these ones that would be considered pro-inflammatory are downregulated. So take-home point is in the MPTP exercise in white, we're seeing clear evidence that these cells coming in are not bad. These cells that are coming in with exercise are providing some trophic support. Okay. So I'm going to end right there with some take-home points that we talked about at this seminar, and then I'll also talk very briefly what we're planning to do, where we're going from here. But basically the idea is absolutely multiple types of exercise seem to be important, but maybe for different reasons. So I think the idea that obviously we're still interested in aerobic exercise, but skill exercise still plays a role here. And it may be that skilled exercise may be tapping into more motor aspects of movement, if you will, and that aerobic may be more cognitive. I think the other thing, when we talk about looking for evidence for plasticity, I hope I tried to convince you that, yes, I mean, we do think whatever the effects of these exercises are in Parkinson's, we very much think that it's happening within a circuit. And again, not to dismiss the importance of dopamine, but we do feel that with the animal data, it's strongly supporting synaptic restructuring and both morphologic and functional changes that we're seeing. In terms of what's driving this, these are studies that are ongoing right now. We are currently looking at a number of different immune factors in a big DoD study that's going on right now, Department of Defense. This is actually with Jack Van Horn and Siva Venkatesh, who are helping us to look at structural and functional changes in imaging over an 18-month period, baseline and at 18 months. And we're also looking to see how this relates to some immune factors that we've looked at in animals. We're going to see how that relates to the humans. We're also looking for changes in a number of neurotrophic factors as well. So I'm going to end with this slide. I just want to remind you, it takes a village. I'm fortunate, very fortunate, that I get to work with a lot of really great people. This is a big list. And again, we just joined is Siva and Jack Van Horn, Dr. Van Horn, which we're thrilled to partner with at University of Virginia. And then Michael Jackwick, a PI, co-PI here at USC, Don Scheisser at UCSD, Dan Holschneider, who I mentioned as well, who's basically here at USC, a big co-PI of this study, and Brett Lund here with a lot of the immune data. Also Adam Lundquist, who did a lot of the work with the astrocytes, has been particularly important. And we have another grad student, Aaron Donahue, who will be helping quite a bit with the immune factors. I thank you so much for allowing me to present this. As I said, I work with an amazing team, and just to emphasize again, it's all about finding a great team to work with. I can't emphasize that enough. And in any event, I'm happy to take any questions.
Thank you so much, Giselle. That was a fantastic tour de force of all the current thinking in Parkinson's disease research. A couple of questions. So I was really intrigued. I've always been intrigued about the role of the thalamus here. It is the sort of the router of the brain, and given the emphasis you put on circuitry, I'm kind of curious a little more about the thalamus, whether or not these are, what are these nuclei that are the ones that you're focused on, or the ones that like the results have borne out? Do these tend to be more association nuclei, relay nuclei? Are they interlaminar? Do you have a sense?
Right now, not really. We actually, that data was actually a surprise to us. We have not really paid attention too much of the thalamus. It was because of the findings that we just showed you, and then looking back at some of the work that Daniel Holschneider did, that we began to see, oh my gosh, there's a lot there that we need to start looking at. I think people certainly appreciate the role more so now, although Mike would argue that people have been looking at it for a number of years, but its role in cognition, I would still say, is relatively new. There's not that much out there on it. So, all those are all great questions. We simply don't know. I can say, though, that, again, if you look at some of the imaging data out there, not a lot of functional imaging, but structural imaging, that there are definitely reports of at least volume loss there, and both regionally and globally in the thalamus. I would also say that, prior to most of the work in thalamus, as you may already know, has really been focused on motor. So again, cognition is going to be kind of relatively new and not that much out there. So I think hopefully you and Siva are going to help us with that, answer that question.
Right. One thing which you touched on it in a couple of different places in your talk is that BDNF appears to have some direct relationship to exercise, GDNF as well, and other neurotrophic factors. And I'm curious about their role in cognition, kind of more specifically, but then also how specific are some of these effects in Parkinson's disease, and are there other implications for other neurodegenerative disorders?
Yeah, I think, again, that is a great question. I think, first of all, people have looked at BDNF in Parkinson's models and have definitely seen some changes in BDNF expression within the striatum. I think one of the questions that often comes up is how relevant is BDNF expression in the periphery, particularly just measuring it. And I think that's a little bit why we got interested in looking at cells. Because the idea is that BDNF may not cross, and but for example, immune cells may, and that may be the more important driver. I think the other thing is what cells may be making locally that may be important here as well. And again, that's the astrocyte and the microglia, both of which could contribute to some neurotrophic factor support. I think the question, though, that comes up is, kind of how regionally specific are these effects? We don't know. We'd like to think that they are regionally specific. We'd like to think that some of this may be circuit specific, maybe because of blood flow, that fundamental changes in angiogenesis is really what's driving these effects. So wherever you're seeing angiogenesis is where most of these effects are happening, we believe. And we believe that because one thing that I didn't get a chance to show was that we are seeing some transcript expression from transcripts that are important in angiogenesis and neurotrophic factor expression that are coming from neurons. So the idea that the neurons that are being activated through the process of whatever you're doing, let's say running or processing information, the idea that neuronal activation may be driving some of these circuit-specific effects. So the periphery isn't creating it; it's the actual use of the brain, how you're using your brain, if you will. The fact that I'm being more cognitively aware of what I'm doing, and again, just to point to the very specific example that I showed you with Daniel, where, clearly these two animals were both aerobically challenged, but the idea that one animal was actually much more engaged can actually change blood circuitry, right? So at least by that particular method, suggests that there may be something about exactly what we said, how I'm using my brain, how engaged I am, that may influence some of these effects.
How transferable are the results you're finding in mice to humans?
That's a great question. So as you can see, I'm a big animal believer, and I often find, it's really all about the question and the limitations of your model and understanding your model quite a bit. I've worked in the model for a very long time. So, we're not in any way, shape, or form saying that this is Parkinson's disease. Yeah. But what I think it does is it really gives us an example of plasticity. And we've certainly been able to demonstrate that in an animal, right? And show that, again, with very little changes in dopamine, you're already getting quite a bit of downstream activation happening. And we've been pretty fortunate in being able to replicate some of these findings. As you can see, for example, D2, we were able to replicate that in humans. And that was the first study of that was in an animal, in the amputee mouse. That's how we learned that. That wasn't a human, that was an animal. The second thing, how we learned, I talked about this increased release of dopamine. That was just replicated by John Stossel at University of British Columbia. He actually used, I think he did use raclopride to demonstrate that there was more dopamine being released, replicating what we first described in the animal. So we're hedging our bets that, it's a good model when you have a very specific question that you want to ask. And then, of course, thinking about how you translate and ask that specific question in humans. But as you know, the study we're doing right now in the DoD is totally because of this animal data, including the immune factors. We're not looking only at we're not just looking at immune factors in the blood, we're looking at cells for the reasons that we talked about, right? The idea that we do think that there could be some of these cells migrating into the brain and looking to see what they're making is also a very important part of this study.
So women are hard to find. Now, is it because there's just a difference in the diagnostic rate of Parkinson's disease in women, or are males just more likely to want to participate in these things? And if that's the case, have you noticed any sex differences that might be between males and females with and without PD?
Yeah, that's a great question. So, in Parkinson's disease, there's a slightly higher incidence of Parkinson's in males, and in Alzheimer's, it's just the reverse. It's slightly higher in women. Honestly, it's pretty common for many studies to get more men than women, although in our current DoD study, we're actually about 50-50. I really worked hard to make sure we got a substantial number of women. In terms of, people have looked at some gender differences between men and women, and there have been some suggestions, for example, maybe men may be a little bit more likelihood for having more problems with the Parkinson's, specifically things like more cognitive issues as an example. But the truth is it's still relatively understudied. So it's not not as much information out there with Parkinson's and women. There's some, but not that much. So hopefully these are some of the questions that we can try to address in our group. I can say that, in looking at some of the, exercise activities, we have women, we've been looking at a lot of different parameters of exercise, including intensity. And, again, this is an exercise group that we're looking at. And I was curious because, we're seeing some really nice data with some intensity and wearables, whether those were all men. And actually, it's about 50-50, men and women. So it's nice to see that our women group are as active as men. I'm not surprised. But interestingly, some other types of exercise parameters that I think will be of interest for us to look at a little bit more closely is back to that VO2 max as an example of cardiorespiratory fitness. One of the things we do know is that a couple of things influence that is our ability to get that fitness level up as measured through VO2 max. Aging will flatten that out a bit, and women tend to be a lot further, just are not able to get up to that level of cardiorespiratory fitness as men do, as measured through VO2 max. So you start seeing some sex differences there on some of these metrics, particularly VO2 max. That's actually known. And we hope to be able to explore that a little bit. I'm also hoping that we're looking at some estrogen replacement questions in this as well. We're going to be following and tracking that as well with cognition. People have looked at that, and there's data both ways, whether or not estrogen is helpful or not helpful in Parkinson's disease for women.
One final thing is, one thing that's striking about your presentation is just the different types of data that you are gathering to look at Parkinson's disease, whether it's, at the molecular level, you're looking at these neurotrophic factors, you're looking at PET imaging, you're looking at, connectivity modeling, across spatial and temporal scales. How important are the computational approaches and the kind of the integration of this data to getting that comprehensive view of Parkinson's disease?
Yeah, I mean, I think that's why you're joining us, Jack. You're going to help us with that, because I think, as you can see, it has kind of this puzzle feel to it, doesn't it? I mean, we've got kind of different ideas of what's happening in terms of the immune, in terms of angiogenesis, even with behavior, and the idea is to try to find a common thread, particularly given the fact that at the end of the day, we are interested in circuit-specific effects. And, trying to think about that spatially as well, where some of these bigger effects are happening. I think that will be particularly important because, again, that will help us guide the animal data in terms of where we really need to be diving down and looking for some of these molecular changes. So, I think a computational approach will help us, without a doubt, really focus in and eliminate things that may be epiphenomenon, but are not nearly as critical as some of these other points. We've talked about that a lot. You get a lot of data, but the question is which ones are the relevant ones, and being able to hone in on those are certainly helped by a computational approach.
Fantastic. And with that, thank you, Giselle Petzinger from University of Southern California. We really appreciate you sharing your work with us today. What an exciting project. Really fantastic talk. Thank you so much. Absolutely. Everybody who's out there listening, thank you so much for joining us today. And we'll look forward to seeing you next week.
Excellent. Thanks so much, guys. You guys take care. Bye-bye now.
Bye-bye.